Welcome to SeqOne's corporate seminar. Thank you all for coming. I'm Jean-Marc Holder, and I'm the Director of Strategic Innovation at SeqOne. Now, we're going to have a very packed schedule today, so I'm not going to talk for too long. But for those of you who don't know SeqOne, we have a cloud-based platform for analysis of genomic clinical information in both somatic and germline. Okay? Now, if you want to know more about what we do, please do come by our booth. I think it's booth 278. But right now, what I would like to do is just quickly introduce the speakers and then stop talking. Oops. Oh, Jesus. Here we go. All right. So we're going to have first a talk. Now, sadly, Michael won't be here today because he had some travel problems, so we'll have a video from Michael. Then Professor Thevenon of Grenoble is going to talk about his experience in rare disease interpretation with phenotyping. Kévin Yauy, who works in the CHU of Montpellier, the Montpellier Public Hospital, is going to talk about his approach to phenotypes and using them in genomic analysis. Geoffroy Delplancq from Versailles is going to talk about pediatric applications. And finally, Laurent MESNARD from the Parisian, the APHP, is going to talk about nephrology. Okay? So we have a very packed schedule. So I'd like to, as much as possible, hold questions to the end. If there's some really desperate question, do go ahead. We'll figure out a way to address it. And then at the end, we'll try to leave as much time as possible for questions and answers, okay? So again, thank you for coming and welcome. And with that, I'm just going to play Michael's presentation.
So that's my pleasure to start this symposium sponsored by SeqOne Genomics. So my name is Michael Blum. I'm the head of research development at SeqOne Genomics. It would have been a pleasure to be with you today, but unfortunately, I was not able to come to Scotland. And so I'm going to introduce this session by giving a short introduction about how to harness artificial intelligence in order to improve the process of a diagnostic with a large-scale next-generation sequencing data. So first of all, you are, I think, familiar with the notion of a diagnostic odyssey for rare diseases. So diagnostic odyssey is this notion that up to 50% of patients with a rare genetic disease have to go through a lengthy process which involves multiple consultation with a specialist in order to have a diagnosis. And so this process is very long, it takes several years, and sometimes there is no diagnosis at all at the end of this odyssey. So it's a pain for the patient, and it's also something which is very costly for the payer. So there is an estimation by the NIH which says that the medical cost related to rare disease is at the same order of magnitude compared to the cost of Alzheimer's disease. And so it's this diagnostic odyssey is clearly problematic for the patient and from the point of view of the payer. So in order to reduce the burden of this diagnostic odyssey for rare diseases, clearly next-generation sequencing data, whole-exome sequence data, whole genome sequence data can have their role to play. So here it's a table which shows like 3 studies which investigated the diagnostic yield which can be obtained with the whole genome sequencing or whole-exome sequencing. So the 2 studies in green are studies related to rare diseases, and both of them, based on whole genome, reach a diagnostic yield of 25%. And there is another study, and I think Laurent MESNARD, professor of nephrology at Paris, is going to mention this study. And it's 2022, there's a typo here. And it's a study related to chronic kidney disease, and it's the same, the diagnostic yield is of the order of 25%. So clearly, whole-exome sequencing, whole genome sequencing can improve the diagnostic yield, and it reaches a diagnostic yield of 25% approximately. And how it works in practice to do the diagnostic yield it's a lengthy process and which involves the interpretation of the variant which have been called following the sequencing of the exome or genome. So here the number which are provided here correspond to number related to whole-exome sequencing. So at the end of the day, when you do the whole-exome sequence, you end up with from 20,000 to 50,000 genomic variants. And then you have to reduce this number of variants in order to find the causal variant, if there is one. And to reduce the number of variants, you do a series of filtering steps. So the first you do quality check, you also consider non-synonymous mutation only, or you remove most of the synonymous mutation in many filtering or pipeline strategy. And you keep only the variant which have a large enough number of reads. And doing that, you end up with around like 5,000 variants. Then you consider only rare variants in a database such as GNOME ID. Because if the variant is too frequent in the population, it can be a variant which is causal for a disease. Otherwise, it would have been purged. And then you end up with from 100 to 500 variants, and then you have to use different diagnostic techniques, different diagnostic strategy in order to find a causal variant if there is one, or in order or if you are unable to find a variant, then you deliver a report stating that you didn't find a variant responsible for the diagnosis. So it's a lengthy process in which you have to interpret a large number of variants. So in order to, so that's a lengthy process, and it's in the future, this process is going to be used again and again, because here it's a report by the NHS in UK, which says that they have an objective of 500,000 whole genome by 2023 or 2024 for several rare disease indications. And it's clear when you read the report that there are like several challenges, and one of the challenges is a very operational challenge. Is once you have the sequencing data, it's okay, but then you need a process that is going to be able to interpret this 500,000 whole genome data produced. So, and in order to address the process of interpretation, they have, like the NHS, provide several recommendations. So one of the recommendations is to hire and to expand the specialist genomic workforce. And the other recommendation is to train additional geneticists and bioinformaticians. So that's what they suggest. But for people involved in diagnostic laboratories today, they know that it's going to be very difficult to be able to diagnose a large number of whole genome, doing like staffing only, because right now it's difficult to staff enough people. So clearly they have to, like diagnostic labs, they have to face challenges in respect to staffing and cost. So it's going to be problematic, and staffing is not going to be the sole answer to address this scaling issue for interpretation. It's one way to cope the problem, but it cannot be the only one. So at SeqOne Genomics, what we do to support geneticists, biologists, and clinicians to interpret the variant, we provide the so-called AI engine, artificial intelligence engine, in order to support variant interpretation. That's the box in red. So what we do at SeqOne, so we don't do wet lab, we don't do sequencing. What we do is once the FASTQ file is obtained by the lab and it's uploaded on our platform, which is a cloud platform, and then we process everything. We do a quality check, primary analysis, secondary analysis. Then we do the interpretation with this artificial intelligence engine. And at the end of the day, we deliver a report for the diagnostic process. And today I'm going to provide several slides about this artificial intelligence engine, and the speaker after me, Laurent MESNARD in nephrology, is going to show what are the results of this artificial intelligence guided interpretation when it's used to interpret a nephrology cohort. So this artificial intelligence engine is called SeqOne Rank Plus, and it relies on two sources of information. So the first source of information is the molecular information. And it's based on the ACMG criteria, which are used to characterize variants. And then there is a machine learning prediction which uses ACMG criteria in order to make a prediction in terms of pathogenicity. So pathogenic variant, likely pathogenic variant, and so on. So it's automatic machine learning based procedure to classify the variant in ACMG classes. And then in addition to the molecular information, we do have clinical information which we use too. And one of the main source of information is correspond to symptoms or phenotype, which are coded using the so-called human phenotype ontology, HPO. So that's a second source of information. So here it's another description of this artificial intelligence SeqOne Rank Plus ranking. So on the top part of the slide, you have the molecular information. So again, we use the SCMG criteria, PVS1, PM5, and so on. And typically to compute these SCMG criteria or features, we use several knowledge databases, such as GNOME AD, for example, but also several databases. And then we have used CleanVar, because in CleanVar, you have a large number of variants which are already classified according to the SCMG classes by expert geneticists. And we use CleanVar in order to train a machine learning model. And in CleanVar, we have shown that the model has 94% of accuracy based on CleanVar. And then there is another layer of information, which is a clinical information, in which we encode the phenotype using HPO terms. And then to capture the relationship between genes and HPO terms, so typically we want to upweight the genes that are associated with the HPO terms which have been provided by the clinician. We use an algorithm called Phenogenus, and Kévin Yauy later during this symposium is going to give you the details about the Phenogenus algorithm. And then we have another step in which we combine the molecular information and the clinical information using, and to do that, we use again supervised learning, supervised by another cohort of around 300 exomes that led to a diagnosis. And then in SeqOne Rank Plus, integrate in a single score these two sources of information, the clinical information and the molecular information. So here it's a screenshot of SeqOne Rank Plus, in which you have molecular and phenotypic information. And just like you see, for example, that the number one variant is a SCMG VUS variant, but it's number one because the genes, the variants here, it's in a gene which is highly specific of the HPO terms which have been provided. Whereas there is a class 5 variant, which is rank number 9, because the gene is less related to the HPO terms which have been provided. So it's great to have a ranking engine, but if I don't even know, in fact, for a biologist or for a geneticist, if there is a causal variant in the exome which I should interpret. And to provide support for that, we have another score, which is called a positivity score for each exome. And the idea is really, it's related to the likelihood that the exome is going to provide a diagnosis. So if it's a large positivity score, there is a large chance that the exome is going to lead to a diagnosis, and that the biologist is going to find the causal variant. And the positivity score, it's related to SeqOne rank score, but it takes the best rank variant, which would fill a list of conditions, which are described below, and which I'm not going to detail now. So that's all for my part. So you are going to have like a next talk after me. So one by Julien Thevenon, who is going to speak about diagnostics of a rare disease and intellectual deficiency based on exome. Then you are going to have Kévin Yauy, who is one of the inventors of Phenogenus, which is the algorithm we use in order to upweight the genes which are related to the phenotypes. Then Geoffroy Delplancq from Versailles is going to show use case of how to use SeqOne platform with HPO terms. And the last speaker will be Laurent MESNARD, professor of nephrology in Paris, and he's going to speak about the impact of a phenotype-driven prioritization for diagnosis in nephrology, and he's going to show an evaluation of SeqOne Rank Plus in a nephrology cohort. So it was a pleasure to give this talk today, and now I leave the floor to the next speaker. Thanks a lot for your attention. So that's...
Can we have the next slide deck, please? Thank you. Julien, would you care to follow up? Okay.
Hello, everyone. So it's very nice to be here in Glasgow, and I would like to thank SeqOne for inviting me, presenting a bit of our work in Grenoble, and for inviting me in Glasgow at least. So first, as a beginning, I would like to thank Claire for the fancy design of my slides. Thank you, Claire. But I need to say that I'm not employed by SeqOne, although the design suggested it. And I have no conflict of interest to declare at all. So briefly, a personal introduction. So I'm a medical geneticist in the University Hospital in Grenoble-Alpes. So I'm the head of the ward of genetics, genomics, and assisted reproductive technologies. And we are hosting a reference center for multiple congenital anomalies for the southeast of France, shared with Lyon, Saint-Étienne, and Clermont-Ferrand. And I'm also involved in the University of Grenoble-Alpes as a coordinator of an INSERM team named Translational Genomics, which hosts a chair, an excellence chair for artificial intelligence applied to health in our multidisciplinary Institute for AI in Grenoble. And finally, I'm also involved in involved in the Oragen Consortium, which I will introduce very briefly because it's not a presentation where I represent Oragen, but I'm speaking from my Grenoble point of view right now. So concerning the aim of the session, the first question that we may want to ask all together is why should we spend time in collecting phenotypes for the diagnostic of patients? So I would like just to introduce a small example of this young girl born from healthy parents, who is a sporadic case developing with a prenatal diagnosis of isolated agenesis of the corpus callosum, confirmed by US and MRI. There was prenatal investigation that were performed, including a normal chromosomal array. And she was born at full terms with parameters slightly on the low side of the growth curve. And she presented at birth a pathological hypotonia and associated with distal arthrogryposis, meaning distal contractures, and morphological peculiarities that you may observe or appreciate on the picture. She further developed with feeding difficulties requiring support, developed frequent respiratory complication, and finally died after a year of hospitalization and medical support that was required. So you want, for the family and for the mourning and for the genetic counseling of the family, you need a diagnostic for such situation. Then you perform an NGS test, which returns a complex information that you don't want to spend time interpreting it. You need then to extract the interesting parts of the raw text into structured language that may be understood by the computer, and that will be developed by Kévin further on just after this. But from this raw text, you may identify human phenotype ontology terms that you can map from the text directly on the ontology. I suspect that everyone in this room knows HPO, so I don't introduce it right now, but we may discuss it later. And you can identify at least a very rich clinical observation with a lot of keywords that you may enrich the observation with. The dysmorphological examination is quite more difficult based on the HPO systematic reading of a text. You may favor image interpretation, either AI-based assisted or directly because you are educated at examining the patients. But what you expect is some magic magic stick that would point to you clinically that this child is strongly suspected of a Boring-Opitz syndrome because you have learned it or because you have collected the symptoms and entered the symptom in an intelligent system that is suggesting the diagnostic, and you want to transform the genetic information into something that is usable and interpretable, understandable for every geneticist, namely a de novo truncating variants in the penultimate exon of A6L1 gene that is responsible molecularly and clinically of this Boring-Opitz syndrome. So what I have tried to exemplify here is that the genomic interpretation is just the correlation of genotype and phenotypes. That's not new. But if you want to generalize and automatize this system in a computer, then you have to collect very standardized phenotypes, so mapping the HPO because it's the most widely diffused ontology and probably the most accurate. You want to list as exhaustively as possible genetic variants. So NGS and whole genome sequencing is a way, but it's not the only way. And you want to collect and structure gene-based knowledge, just as Michael just exemplified before, to have very structured information regarding the molecular mechanism of each gene and associated disorder, each symptom and associated disorders, and its inheritance mode in case of monogenic disorders. And our capacity of interpreting the result is residing in the intersection of these three circles, because you want to explain the phenotype of the patient that you have observed, explained by a variant that you have detected, and a variant that you may be able to classify and interpret because you have acquired knowledge, either individually or structured in the computer. There's no difference in it. So there are two ways of performing these genotype-phenotype correlations. The traditional way, phenotyping firstly the patient and constituting a priori from the genetic test homogeneous groups of patients presenting with the same or comparable phenotypic expression, because we are educated as geneticists to recognize several hundreds of syndromic disorders that we may be good at diagnosing clinically. And then after the clinical examination, you apply the genetic test and confirm the homogeneous genotype-phenotype correlations. But we all know that this approach can be very effective when you are able to recognize the disorder, but it's quite ineffective in heterogeneous disorders such as intellectual disability or autism spectrum disorders, or a lot of disorders that we mostly encounter in clinical routine. In those cases, you want to screen very widely the genome of the patient, acquire the variants, then return back to the phenotype to perform some kind of reverse phenotyping, although I think it's better to collect before a priori of the test the phenotype. And then, if necessary, you can share the information with international collaborators, but the final objective is to constitute a posteriori from the genetic and clinical examination some correlation between genotype and phenotype and constitute homogeneous groups of patients. So now, how to collect phenotypes in routine practice? It's just personal examples, so it's not a recommendation that I don't suggest you to do the same. There are some problems with our choices, but I'm quite happy with it. We have installed several years ago a phenotype server instance in the University Hospital of Grenoble, so we are autonomous in collecting the phenotypes of the patient. So this is an example of a clinical observation with the family history that is drawn on a pedigree, with some growth charts that are readily available in the application, and the HPO terms that are translated in French. And I would like to thank the resident in medical genetics in France, because every one of us have contributed to this translation several years ago. And everyone who knows phenotypes knows that they have a suggestion system for clinical diagnostic and for gene panels that may be used, exported, or applied to some test. So the open source phenotypes application is not maintained as far as I know, because now it's a commercial usage that is promoted, but I think that the open source old school GitHub depot is still available and functional. And we also have an electronic health record, which is named Easily. and obviously I have to say that it's not that easy to use, but it has the advantage to gather the clinical symptoms in structured formulas directly in the clinical routine when you meet the patient, when you do the follow-up meeting with the patient, or the clinical review of the file. So it's quite efficient to have everything centralized in one system. So it's the way we are promoting the system now, because it's quite confusing for all the colleagues to use multiple softwares, because we have so many softwares to use and they are never communicating. So we are focusing on this easily software. It's a regional software. But the main point is here, concentrate the efforts in one software independently of their limitations. And on both applications, we can export HPO terms that are either like in an informatic format, like a JSON file exported from the phenotypes application, or we have been able to ask for interoperability modules that can navigate the clinical information from one system to the other, and then to directly fill the software that we are using downstream the clinical capture of the patient phenotype. So how we integrate this phenotype in interpretation or genotype-phenotype correlation, I would like to just exemplify two examples. So it's not the only way we are thinking in the diagnostic work in Grenoble. We are quite more subtle, hopefully. But macroscopically, we have two genome-wide sequencing approaches, one which is time-effective for specific occasions, such as prenatal diagnostics, and the other one who is less in the long time of the diagnostic through a regional platform for whole genome sequencing. So just to illustrate how SeqOne is positioned in this local organization for our fast track exome sequencing process. It's not that fast track because you see the turnaround time is about 4 weeks, but 4 weeks is a reasonable time, I think, for coordinating the appointments after the diagnostic of a fetal anomaly that has to be confirmed by a referring echographist, performing the sample of the amniotic fluid, extracting the DNA, sequencing the samples, pushing the data to the SeqOne cloud application, interpreting the data in the week that they are produced, and then another cycle to validate by an orthogonal approach the variant that is candidate for the diagnostic. We still use Sanger sequencing for these applications, and especially for a genetic counseling in prenatal period. We want to be sure that this variant is true positive. And in the 4 weeks, we are mostly, not in every case, able to return back the result to the family. We have chosen to rely on SeqOne Cloud for this application because we have a very limited flux. So it's the very transparent account of every whole exome sequencing that we've performed locally in the antenatal period. And you can see that it's been starting very slowly, and we had then the agreement for funding more exams, and now we are performing 3 to 4 trio-based whole exome sequencing, 2 per month. So it's like 8 families that are pursuing this pathway. It was too limited flux to involve too much energy in setting up a bioinformatics infrastructure locally, because you need to consider a lot of details that you need to take time to consider, and it was impossible with our human power that was present at the time. So we are still happy to work with SeqOne Cloud for this application, and also because the biologists that are interpreting the results need some ergonomy and some easy to use interface. And SeqOne was offering readily this application. So I will not detail the clinical indication and pregnancy outcomes of the whole exome sequencing that we've performed for time limitation reasons, but we can discuss it later on. But just like in most centers, cardiac malformation, brain malformation, or multiple congenital anomalies are representing most of the cases that we are prescribing. And in most cases, as you can see, the pregnancies can follow after the whole exome sequencing, so that's an important thing to notice, but I'm not the only one to say it. One thing to mention also, although, is that we are prescribing whole exome sequencing when the diagnostic or the prognostic is not certain. So if the family has decided to go for an termination of the pregnancy, then we do not propose whole exome sequencing. So it's restrictedly proposed for the difficult situation for the families. So it's a quite psychological consuming application in the service. But so this is for the limited flux of analysis, and we need to scale up the flux because everyone knows that rare disorders is quite frequent overall. So for scaling up the process, I will just make a turn for our French National Plan for Genomic Medicine. So you have the URLs if you want to look for. There's an English version of the website, which is supposed for the 2025 year propose healthcare pathway for patients with rare disorders that have no clear diagnostic, and they can benefit from a whole genome sequencing that is realized, interpreted, and returned to the patient in the clinical and diagnostic framework. And we have been selected as a regional project, Oragen, for being one of the 2 sequencing platforms in France so far. So we hope that there will be more of us soon. And in Lyon, all the sequencers are. So the samples are gathered in Lyon, and all the data for germline sequencing bioinformatics goes in Grenoble and is handled and managed by the team. So we have a bioinformatic team, so it's an assumed choice to have SeqOne involved in the prenatal setting. And all the south of France is involved in prescribing the tests, and all the south of France is involved in interpreting and returning the results. So we hope that we reach our goal to gather everyone that is motivated to participate to this quite big adventure. And it's building and learning as working, let's say. We have developed a bioinformatic dedicated for each family structures. We have pipeline devoted to each kind of prescription, mostly trio-based sequencing, but sometimes you miss one parent, or sometimes you have only the child that is sequenced, or the adult missing these parents for age-related reasons. Or we can accept more complex family structures. We have developed an in-house interactive and integrative automated report, so that is proposed for each file, each prescription, and resuming the clinical information that is collected, the variants that were proposed and pre-qualified, and allows a fast interpretation for easy prescriptions. Just to give just a quick glance to this report, we have an increasing proportion of the reports that lead to a variant that is suspected to be causative that are returned to the patients. And here you have represented the percentage of reported variants that were automatically prioritized in our automatic reports. So at the beginning, it was 2018, it was 92% of all the pathogenic and likely pathogenic variants that were reported. And you see that we have ramped up to reach, it's been 6 months now that we are just slightly progressing from 99.2, 99.3%. So we are not reaching the 100% that are concentrated in our automated report, but I hope that we will not reach 100% because we need to face the complexity. And this is the position of SeqOne in our framework, because you need to go further than these automatically screened variants for performing some complex diagnostics. And this is offered by the platform SeqOne because of its ergonomy and its ability to personalize the filters that are available through the interface. So a quick word on the positioning of SeqOne in our framework. So we are mastering internally the pipelines. So that's an original implementation of SeqOne that is not supposed to be reproduced. And we are using open sources visualization software, including IGV and others. And we use SeqOne as a variant interpreter interface. So we push the data that we are manipulating to the interface. So it's been a huge work of all the SeqOne team, and I would like to thank them here officially. And it was necessary for us to include them this way because we needed to have the way back to the clinical report that is issued in a different information system. So everything is focusing in this automated report that is positioned at the center of the slide that is in French. I'm sorry for this. So as a conclusion, and to wrap up the main ideas that I mentioned, the benefits for me of this collaboration with SeqOne is that they are proposing an easy and user-friendly interface that is suitable for a broad usage in genetic diagnosis. For small-scale projects or in-house local sequencing projects, they can provide a ready-to-use platform hosted in the cloud that is very easy to set up and to start to work with. And the main application that we are using SeqOne with the Oragen group is that we benefit from their very nice customer support or user support, because we are facing something like 180 biologists that are participating to the Oragen consortium. And although I can speak to you quite easily, I cannot speak to everyone very nicely. So it was necessary for us to have a professional customer support. And so this is one of the main expectation that we have for SeqOne in Oragen, besides this variant interpretation interface. So I would like to thank you for your attention. Thank also all the Oragen team, prescribers and interpreters, the local ward. And I need to say that I am recruiting a biologist or someone that is liking bioinformatics and biology, or a geneticist, at least someone nice and good.
Thank you very much, Julien.
Thank you.
As I said, we'll hold the questions to the end, and I'll basically ask Kévin to come right up, and we'll follow on from there. Thank you.
Hi everybody, thanks for attending to this presentation. Thank you SeqOne to invited me to present to you this work that I've performed during my PhD, talking about how can we use phenotypes and AI to improve diagnostic. So I'm Kévin, I'm an MD in medical genetics and now PhD in bioinformatics and data science. And so I'm happy to present to you this work. So I will not go further into this. We already said that rare diseases affect a lot of people, 3 million in France, and the diagnostic challenge is something difficult, especially because you have a lot of different rare diseases that is known. And as I like to say, as a young geneticist, I have only one brain, and it's kind of difficult to learn all these different rare diseases. Even because of our job, we don't have any choices than deal with a lot of different rare diseases. So since a long time ago now, all of our community started to think about the computational phenotype analysis. Before there was like POSUM, stuff like this. I don't know if you know, London database, stuff like this that was helping us to browse into the knowledge of rare diseases. And how does it work? We perform phenotyping, so we are looking for symptoms that is different from the normal. And we use ontology that allows the human and machine to discuss together in the same language. And I think now everybody is using the HPO, the Human Phenotype Ontology, which is an ontology that is constructed as a human development ontology. If you have this, then we may achieve AI system that help us to browse the knowledge and help us to find the correct diagnosis. So problem is with phenotype is, as it said here, is reported to be fuzzy. I think we kind of all know now that when you ask physician to extract HPO term, it's kind of not always the same. We do act different each other. So phenotyping can be fuzzy because either we have it's an heterogeneity in phenotyping, either we have variety in expression for clinical symptom in each diseases, or we can have undescribed association. That's because we already never discovered or described this. So this is a hypothesis, but actually there were no really studies that try to see how, as medical doctors, we phenotype a patient. So we performed this analysis. We gathered 1,700 patients with HPO term that was provided and the correct diagnostic in gene. And what you can see in this weird tree map is that you can see that there's a lot, each rectangle here is a symptom, and there's a lot of different symptoms that was inside this cohort. Actually, half of them was only declared only once. So it's really like, it seems that it's difficult to use HPO term at some point because all the patient has different HPO term at the end. So this heterogeneity was something that we were struggling, either because maybe physician phenotype differently, or because maybe it's just like patients are different. So we were asking ourselves, is it because of one on the other? We asked then, we made a small experiment, we asked some physicians, 12 of them, to phenotype the same patient. So we have 3 same patients, and we ask physician, can you provide us the HPO term for this clinical cases? And I show you an example here that see that we perform really differently. It's really because as doctors, we don't phenotype the same way. So as you can see here, there's a lot of differences between this physician in the number of symptoms that is provided to us, but also it's different in the selection of the HPO that they provided to us, because at least there's no one HPO that was provided by all the physicians for clinical cases. So yes, physicians do phenotype differently. So the question is now, okay, if physicians phenotype differently, does it match with the knowledge that we can access online? And the answer is like half of them are not existing in the database that there is online. So it means that this phenotypic heterogeneity that exists, because we do different as a physician, it's not matching what we have as a knowledge online. So we were thinking about how can we try to go to 100%? How can we try to fill the gaps with AI? So let's take like a brief retrospective of how we do. So as a physician, you learn a lot of different cognitive frameworks to solve problems. Here is an example. You have a child who are not able to walk at 18 months. So you can either say that this child has a motor delay. You can say, oh, because he has an hypotonia that explain why he's not able to walk. We have different ways to say the same thing. But the truth is that we are going to select only one term of this. We're not going to put everything. And this is kind of difficult for AI because it cannot guess that you wanted to say the same thing at some point. Or you have to say, or you have to declare it. So as you can see here, these 3 different terms have a lot of gene in commons. That's not surprising, of course, as you may know that this are symptoms that is related. But as you can see in the ontology, these symptoms are really far. They're not really close. They're even like the maximum of difference. And so human phenotype ontology is not made to link symptoms each other. So we were thinking about how can we, how do medical physicians think? I think we all do this kind of reasoning, which is called the inductive reasoning. If you see microcephaly and a delayed ability to walk, then you think about all the disease that you know, and you may think about, okay, if the patient has intellectual disability, this is probable. This is something that we do. How can we use AI to do the same thing? Well, I think we all have used in your life maybe once Netflix. You may know that they are really efficient to suggest to you some movies. And what how do they do? They think about, okay, if you want to provide a recommendation to your child, saying that what kind of movie he want to watch, you already know what kind of movie he like, and then you think about, okay, maybe this one could be nice. So we do this all reasoning every time. And it exists some model that do the same. So what we did is like we gathered as much as we can HPO gene association. We gathered a lot of them, and we created a system that is able with AI to kind of reproduce this. So it's a weird word. It's called non-negative matrix factorization. It's an AI system that tries to reduce the dimension of a lot of different symptoms existing into groups of symptoms that is related to the knowledge that we know for genetic diseases. So we perform this and we get the best of it. And it looks like this. It's the universe of symptoms. There's symptoms that belongs only to one branch of the HPO ontology, but there are a lot of them that is interacting each other. It's not surprising, but this is how it looks. So using this, we were able to compute new symptom gene association that was not available, a lot of them, and using that, we are able to match the heterogeneity that physicians have. So it was nice. This is an example. Yeah, this is the autoimmunity group. In the autoimmunity group, you have a lot of symptoms, and these symptoms, as you see, they are all related. You're not surprised to see that autoimmunity, vasculitis, arthritis, all are close in the relativeness of diseases. And this is all AI computed. He can learn this and show this kind of pattern, this kind of group. Using this, compared to state-of-the-art methods, provides really better performances just because at the end, we just deal better with heterogeneity of physician. If we compare with other tools like called PHRank, PhenoApt, Phentogene, all this, Okada, this is the tools that was published on 2022. We have, as you can see here on the green line, we have better performances in the ranking of diagnostic gene because we deal better with heterogeneity of physician. As already mentioned, so we implemented this into the SeqOne Rank Plus, providing information about the clinical symptom of the patient and helping us to have interpretation of the variant. This providing HPO term provide really a better insight and a better prioritization, so I will not go further into this because I know that Laurent will provide some insight on it. And to conclude, we provide this tool which is called Phenogenius as open source, so everybody can use it. This system is available as preprint and mid-archive, and of course it's available in SeqOne Rank Plus. So if you want to know more about it, don't hesitate to come to ask me if you need more details, and I wish that this system implemented will help us to facilitate interpretation of exome and genome sequencing.
So thanks. Do you hear me? Thanks to invite me to present on everyday life how we use HPO in clinic and in neuropediatric and prenatal diagnosis. So I have no conflict of interest to declare. We just use SeqOne when I arrived in Versailles, where SeqOne was already there. So So just to come back about the human phenotype ontology, it's work like it has a tree structure, as you see. So you have the main, you have the trunk of tree, and that after that you have branch. So, for example, you have neurodevelopmental abnormality. Then when you focus on that branch, you have intellectual disability, specific learning disability, and when you go to the smaller, very smaller branch, you will have more specific HPO terms, so more very variant, for the variant prioritization. If you go deeper on the HPO term, you won't have the same gene. So for me, that's important to have that in mind. So we go on a clinical case. So we saw that patient, the couple, with for trophoblast biopsy, because there was a risk of trisomy of Down syndrome actually, and an increased nuchal translucency. So we perform a karyotype in array CGR that were normal. And then after that, the follow-up, we revealed that there was an agenesia of the corpus callosum and a doubt about ventricular septal defect. So now we have two HPO terms then. And so we decided to perform trio-based exome sequencing. And during the pregnancy, we did 3 interpretations because we are new HPO terms were revealed during the follow-up. So we first do the interpretation with that term, increased nuchal translucency and agenesia of the corpus callosum. And what we will also, I will switch to the internet presentation. So here you have the platform, actually, as you can see. And so I have implemented some my HPO terms. So increased nuchal translucency, and the other one, genesis of corpus callosum. And it's revealed that it's still we have a variant of interest and NRAS. As you know, it's involved in Noonan syndrome. Go back to the presentation. After that, there was RMI was performed, and it's confirmed the agenesia of the corpus callosum, and also cardiac ultrasound confirmed the ventricular septal defect suspected. So we redo an interpretation of that, and we keep the NRAS variants. And then we perform, during the ultrasound follow-up, it's rather an aside, so we were thinking about a metabolic disease, but we did not remain any other variants. And that really ended the end of the pregnancy. Cardiomegaly and hypospadias were revealed. So despite of that, so we don't need that anymore. So we keep that variant, but still a VOS, because it's not reported, as you can see here, in a GNOMAD database V2 or V3 version. The variant is not reported in ClinVar, neither in LOVD. But despite there is quite bioinformatic score, they are quite, it predicts the variant as damaging. And all these kind of screenshots were obtained from Moby Details. I have no conflict interest with them, but I really recommend you to use it because it's very friendly and you have all the data to do your variant interpretation. And you can put the classification also of your variant, and you can be contacted by a colleague. It's very user-friendly, really. So after that, so the baby was born. So neurologically, we have the agenesia of the corpus callosum. A new cardiac ultrasound was performed, revealed a biventricular hypertrophy, which can be a sign of the Noonan syndrome. And so we have clinically, we have, so as you can see here, down-slanted palpebral fissure there, but with the edema it's not very clear. But for sure, here you have low set hairs, and there is also umbilical hernia. And that's a picture that quite really trendy, maybe, you know, phase 2 gene. I don't know if you use it in your everyday practice. Not personally, but sometimes it can be useful, and it seems that there is a match with the eye of the mother. So we go back to the internet presentation, please. As we can see, so we have the NRAS variant here, and if we put our HPO term, the postnatal one, with cryptorchidism also. There, and there is non-latent palpebral fissure. You see that our variant with only 3 or 2 term HPO term, we can have the prioritization of the variant. So back to the presentation. So what I wanted to say that's been very well, we phenotype the patient quite different in our practice, but we can have a genotype without a good phenotype. And I think that HPO, and I'm really convinced, and I hope I will convince you that HPO is the way to describe the phenotype, for sure. HPO significantly improved the variant prioritization, but 3 or 5 HPO variants are usually sufficient to prioritize your variant and to find a diagnosis or a candidate genes. And the more specific the HPO term is, the better is the way to prioritize the variants. But you have to also be careful about that. If you go very deep on the branch, you will have a very specific HPO term, and you can miss sometime candidate genes. So this is why it's always good to have more HPO term, but I don't know in your practice, but 5, that my point, 3 or 5, is that quite that's sufficient. And despite of that, we have I really think that may be a diagnosis, but it's still a vote. So the chain we find need further validation. Yeah. So thank you for the other clinician of my department who saw the patient. Also the gynecologist department and also the pediatrician will follow the kids. And like Julien Thevenon, we are also hearing, but specifically a clinician. So you are welcome to Versailles. For sure you have to speak French, but you can speak French with a Scottish accent. It's totally perfect for us. Thank you.
Going to be Laurent, and he's going to talk about our applications in nephrology, and then after that we'll take questions from the audience and from the net. Laurent, over to you.
Hello everyone, thank you for the invitation. I'm willing to convince you that nephrology, and especially the field of adult nephrology, is very play a meaningful role to use the HPO term. So just a brief overview who I am. I'm as well the national coordinator for rare disease of the kidney in France, and I'm associated with Julien in the PFMG 2025. And I'm very fan of genomics and bioinformatics, and currently at Sorbonne University, we are doing almost 1,600 whole-exome sequencing for chronic kidney disease, mainly of unknown origin. So just to give you an overview, I'm working on in Paris. We are one of the top ranking center in France in the specialty, and we are doing 30% of the test during the hospitalization. It means that we are providing the test when the patient enters our center for the very first time for a long hospitalization for the entry point of your disease. We have currently 3 physicians trained in nephrogenomics and 1 genetic counselor dedicated to this activity. We started up in 2019. And as we structure during the very beginning of the process, during the prescription is the clinician who is collecting the HPO term using a survey with a red cap format. And one of the specialty we did develop is what we call co-interpretation, that the clinician is participating using the SeqOne platform to the interpretation with the molecular biologist. So this is the numbers we are doing in our center. So to go a much broader view of nephrology, adult nephrology, this is the cost of the specialty. The larger the disc, the large number of patients, and you see we are basically a very few number of patients, but the cost in abscess is very high. Basically, the nephrology engulfs more than 4 billion euros per year.
And the number of patients are quite that high, 80,000 patients. Half of them basically are kidney transplant and the rest on dialysis with a mortality rate quite elevated. And one of the questions arising is when we can start a genomic test in those populations, because in this population, we do not know currently that 10 to 30% of them, those patients are suffering from Mendelian disease, especially in the subgroup of unknown nephropathies. And we do observe that 30% of patients do have a family history of end-stage kidney disease. I'm not talking about hypertension. It's a very hard disease, I say, with renal failure. And we not discuss the impact of what we can do with exome sequencing in kidney transplantation, comparing the recipient and the donors. And the thing it's very important is not to make a diagnosis for the index case, it's to provide this information, all the relative. Because we in nephrology, we have the possibility to provide therapeutic that could limit and retard, delay the entry point of end-stage kidney stage. So the year off of dialysis could be a very high value for the patient in terms of mobility, but a very high value for the society because you can delay the entry point in dialysis, which is very expensive for the patient. And so you can detect early on the relatives and provide a cascade strategy, cascade screening to organize the prevention. So I think all exome sequencing, all genome sequencing, and genomics in the field of nephrology are very high value for prevention strategy at the national level. This is just a cartoon of the diagnostic utility of all exome sequencing in nephrology. There is a seminal paper published by our colleague of Columbia University in New York showing that in more than 3,000 people, we can have a diagnostic yield of just, I would say, 10%. But in this population, there is no selection. Just take patients entering in dialysis from, I would say, 18 to 99-year-olds. With those patients could suffer from diabetes, immunologically related disorder, but not special things that could be Mendelian related. In the other end, in the pediatric population, the diagnostic is much higher, 32%. And in the between, all study, I will show more data on that, provide 24% of diagnostic yield in all those patients in a sample size currently of 500 or more patients. And in those 500 on patients, we did publish that last year. We did observe that 26% of Alport syndrome, 6% of autosomal dominant tuberous cell kidney disease, 21% on ciliopathy, and most importantly, 33% of the diagnosis are linked to the category of other. It means one specific gene to one specific condition. It's very hard to predict what is going on at the very beginning of the process. And in this framework, we do provide 34% of complementary targeted sequencing in relatives or in the patient to rank the variant to class 4 SCMG. So in the same assay, we are able to provide copy number variant information, and it add a little more of positive diagnosis in PHP1, Claudin 16, and other gene listed here. It's a tiny percentage, but very meaningful. And so currently, we move more. We're adding more 1,000 patients, and basically the numbers stay the same. So we are stick to the 25% of diagnostic yield in both populations, even though we increase the entry point currently. And there is a very large heterogeneity of clinical presentation. Here on the left, this is the clinical category of congenital or cystic disease, glomerular disease, tubulointerstitial disease, unknown nephropathies. The physician could not know just small kidneys and hypertensions. Or sometimes we do have a kidney biopsy that could let us more glomerular or vascular. And at the end of the story, when the molecular proof is given, you see that even though we are under basalopathy called 4A-related nephropathy called Alport syndrome, which should be glomerular itself, it's very heterogeneous. The only category with not heterogeneous is autosomal kidney disease with cyst. So at the very beginning, it's very hard in nephrology to say this panel will lead to diagnosis because the diagnosis is very heterogeneous, it's misleading. And our colleague from Italy provided the same data recently, showing that at the beginning, the clinical category is very fuzzy and led to different entities at the very end with a molecular proof. This work is very interesting because they provide that the early prescription or whole-exome sequencing will limit the cost of the charge of the patient for the society beyond providing a large number of diagnoses. So just go back to one peculiarity of a center is what we call the co-interpretation. So this is the clinician who will make the prescription, who structure the data at the very beginning of the process, and the molecular geneticist after the sequencing will be co-interpreting the data together using the SeqOne platform. SeqOne provide finally a viatic for discussion between the two physicians, and this will lead to very good phenotype. So on this framework, we also used the AI-powered software offering by SeqOne to prioritize our variations. And we just discussed in the few minutes the interest of SeqOne offerings in nephrology. So we start from a subset of roughly 1,000 exomes among the 60,000 currently sequenced. And we do have 242 positive exomes for which we did find a causal variant, and the rest was so far and today currently negative. And this is just a representation on the number of HPO we did collect at the very beginning during the prescription. And this is the top 10, and to show that we collect in basically at least 6 to 10 HPO term during the prescription. And so HPO term did improve a lot of the diagnostic yield using SeqOne Plus, as we gained from 37 no HPO to 66% with HPO. And the causal variant was on the first page using SeqOne Plus. So we also look at the negative versus the positive exome, and we set a threshold for positivity, arbitrary threshold of 50%. Using this threshold, you are able to catch, using AI, automatically 80% of positive diagnosis, and you can miss 13% of diagnosis, but you can play with the threshold for positivity. So we can examine the negative exome and look at back if you did miss a diagnosis, and indeed it's yes. We miss 7 diagnoses that will be catch later on by AI and the SeqOne Rank platform. So at the end of the talk, we can go deeper in phenotyping. One way we are experiencing at the moment in Sorbonne University is to record the consultations, translate the consultation from speech to text, and provide anonymization. And using GPT-4 or model, we can catch the HPO from the text and use those HPO in the SeqOne Rank platform to accelerate the diagnostic. So my feeling is that we need HPO from the electronic health records, from speech, from anywhere we can catch them to increase the yield of diagnosis and automate the process. I think the model of co-interpretation will on the top provide more HPO during the discussion with the clinicians, advocate for a full AI workflow that will accelerate diagnosis rate and the turnaround time. Thank you for your attention.
Okay, thank you very much, Laurent. So basically, that's it for the formal presentations, and we have a little bit of time left if there are any questions. Yes, I was just saying that we've finished our formal presentations and can now take questions if there are any. And also, people who are signing in from remotely, feel free to ask questions through the chat. So are there any questions? Yes, over there. Claire will bring you a microphone. And then I guess, perhaps depending on who it is.
It's on?
you can go to the podium to answer the question.
Hi, this is a question for Professor Thevenon. If I recall right, you used the Frank system for the report on Oragen. And I was wondering, we saw on Kevin's presentation that the phenogenesis is better than the Frank score. And I was wondering if you're going to implement it into the rapport.
Thank you for the very precise question. That is interesting. Very restricted number of persons here, but yes, we have changed Frank phenotype ranking, which was a very quite effective software for Phenogeneus because it's better. So it's been something like 6 months that Phenogeneus is included in every available report, so you can fully interpret everything on Origin.
Next question. Are there any questions from the online audience? No? Okay, well, listen, I don't think there's any more presentations here. So if anybody wants to come up and talk to either our speakers or to the SeqOne team, we'll be around. So we'd love to speak with you. And if not, thank you very much for coming. It was really a pleasure to see all these people here. And, well, all the best in your further endeavors and have a great show. Thank you.